So I will give a brief talk on different methods for studying dentate gyrus circuits. And I'm going to be talking about adult neurogenesis, and I'm going to give a really brief introduction on adult neurogenesis, but I'm going to really be focusing on the techniques right now. And the main, let's say, background and the main story on adult neurogenesis is going to be there. So the second half of this talk is going to be on Friday afternoon at the symposium. So you probably, if you are interested, you might want to go there. I will start by these two papers that sort of made a discovery initially of this channel rhodopsin 1A, and it was discovered as a ligand-gated proton channel in green algae. This was in 2002 by Peter Hegemann. And basically what this paper shows is that there's a channel that is present in algae, and this channel is light activated, and it will open a pore that is going to allow protons to go through. And it's a very interesting discovery. It was a science paper, but it was the first step towards a technique that was going to really change neuroscience in the following decades. 2 years later, channel rhodopsin 2 was discovered, and this was a slightly more interesting channel from the point of view of the technique. This channel was already cation selective, so it's a sodium-potassium channel, and this was also present in algae. And in this paper, they showed that using channel rhodopsin, by expressing channel rhodopsin in cells, basically, in the final conclusion of the abstract, they say, finally, we demonstrate that channel rhodopsin 2 may be used to depolarize small or large cells simply by illumination. And I think this is going to be the first step towards what we are actually witnessing right now, which is a revolution that is pushing neuroscience towards limits that probably a decade before or 15 years ago we didn't really think of. I'm not going to show you the limits of the technique. I'm going to show you basically some experiments, but probably you will see towards the different talks, especially with Laszlo and with Mike Heuser, that this can be pushed all the way towards controlling neuronal activity in behaving animals, and this is really powerful technique. This is the first paper by Carl Diesel group showing that by expressing channel rhodopsin in neuronal cultures and flashing light in the cultures, you can have an inward current in the neurons, and an inward current in voltage clamp is going to be converted into a depolarizing current in current clamp, and by flashing a light, you can have neuron spiking, okay? And this is the first step towards controlling neuronal activity. And at the same time, there was another paper by Stefan Herlitz showing basically the same, expressing heterologous expression of channel rhodopsin in hippocampal culture neurons, showing that by expressing this current, you can flash a blue light in a neuron, and by flashing a blue light, you can have depolarizations that can be suprathreshold depolarization or subthreshold depolarizations. So by really brief pulses of blue light, you can control the activity of many cells in a dish, or in an animal, or in the system that you're interested. There are different proteins right now, and this is showing different types. This is channel rhodopsin, which is a cation channel that is opened by 470 nanometer lights. Then there is this other channel that is also cation channel, and it has a different wavelength sensitivity. Then there is halorhodopsin, which is a chloride transporter, and the chloride transporter is going to hyperpolarize the cell, which means that now you have tools that are going to be used for activating neurons and also tools for silencing neurons. And right now, this is from a paper by Carl Ditheroth a few years ago, showing that there are many different modifications of channel rhodopsin that are changing the current properties. So you have an inward current, and as usual, inward currents are going to be depolarizing for the neurons. This is the initial channel rhodopsin that shows that the inward current is a peak inward current, and there is a fast inactivation. And through mutations in different subunits of this channel rhodopsin gene, then you can get different properties, such as eliminating completely the inactivation, or having faster inactivation, or having different kinetics, and these kinetics are going to be converted in currents that are going to respond differently to brief pulses. So ultimately what you want is to be able to control a neuron with a really brief pulse, and that neuron, what you really aim is for that neuron to be active only at the moment you give a pulse, and to control presumably the neuron to a very high frequency of pulses. So by giving different frequencies from 5 Hertz, for example, to 60 Hertz of blue light pulses, what you like ideally is that the neuron will be able to follow each single pulse with a spike. And here you can see that different channels with different kinetics are going to bring you different responses. For example, here, if you have a channel that it inactivates to a depolarizing level, which is not 0, when you give a light pulse, then you are going to have a response here, which is a brief barrage of action potentials, but then the neuron is not going to be able to follow the train. But if you have really fast tools, these neurons are going to be able to follow to really high frequencies. So right now, there is a really huge variety of different flavors of channel rhodopsin for activating neurons, allorhodopsin or archrhodopsin for inhibiting neurons, that are allowing the control of specific neuronal populations in different systems. Could be in culture, could be in vivo, or could be in slices. Why are we using optogenetic tools? So what we are interested is in what's happening in the dentate gyrus of the hippocampus, and briefly, you probably all know that the dentate gyrus of the hippocampus is generating neurons throughout life, and in all mammals there is generation of dentate granule cells in the dentate gyrus of the hippocampus. The dentate gyrus of the hippocampus, you probably all know, receives inputs from the entorhinal cortex and sends outputs towards CA3 pyramidal cells, and CA3 pyramidal cells send their outputs to CA1 pyramidal cells, and then this information goes back to the entorhinal cortex. But the main point here is that the generation of new neurons in the dentate gyrus through life, and this is happening from mice to human. This was discovered in the 60s by Robert Altman, and it was not accepted really quickly, mainly due to the fact that it was really not understood. How come the dentate gyrus could generate neurons? What is the origin of these cells? And now we know, and you are going to hear also from different talks today, also including Benedikt, the fact that there are neural stem cells in the dentate gyrus that are able to generate neurons, and these neurons are going to develop and mature during several weeks until they become fully mature. So this is a process that is ongoing in the adult brain, and it's very slow. We are witnessing neuronal development occurring in the dentate gyrus, and the question is, what is the biological relevance of this phenomenon? What are these neurons doing in the dentate gyrus? Why is it important that the dentate gyrus generates cells for hippocampal function? So what is the network that we are looking at? We have the inputs from the entorhinal cortex, which are glutamatergic axons that are coming towards the dentate gyrus. There are dentate granule cells, which are the principal cells of the dentate gyrus. Granule cells are receiving excitatory connections from these axons and sending glutamatergic axons towards CA3 pyramidal cells. And also, in this way, we have lots of GABAergic interneurons and also MOSI cells, which are glutamatergic interneurons, but we're going to focus mainly in GABAergic interneurons. And so here we have GABAergic interneurons that are also receiving inputs from the entorhinal axons and making contacts onto the granule cells in a feed-forward manner. So an action potential coming from the cortex is going to activate dentate granule cells, but it's also going to activate GABAergic interneurons that are going to feed forward inhibition onto dentate granule cells. Then we are going to have also the output of the dentate granule cells, the MOSI fibers, is going to contact GABA interneurons in the hilus, and there are many different types of interneurons, as you just heard, and these interneurons are going to have different functions. Some of them are going to be contacted and they are going to be doing feedback inhibition onto the dentate granule cells, and also there are different types of feedback inhibition. Some of them are going to be doing feedback onto the soma, and some of them onto the dendrites. And there is also interneurons in the CA3 region that are going to be doing feed-forward inhibition onto CA3 pyramidal cells. So as you can see, this sort of simple circuit, which is the excitatory pathway, glutamate coming onto dentate granule cells synapsing onto pyramidal cells, is also highly controlled by GABAergic interneurons. And the question is, how are these cells that are born in the adult brain developing and contacting through this network and making sense of information processing? Okay, so in some studies that we have done, we have studied the input to these cells, and we have shown that the way in which they are connected as they develop follows similar rules as in development, meaning that initially they are contacted by GABAergic axons, then they receive glutamatergic axons. Let me go back one slide. So initially they receive GABAergic contacts in the dendrites, then later they receive glutamatergic contacts in the dendritic spines, and then the final step of maturation is that they begin to receive GABAergic axons onto the soma, the perisomatic inhibition, okay? So, and this happens during a period of about 6 weeks of development. And the next question that we wanted to ask is, okay, that's really fun with the input, what happens to the outputs? Who are the targets of these cells? And to ask who are the targets of these cells, here you can see. So one way of labeling adult-born neurons is to inject a retrovirus that is going to infect dividing progenitor cells, and all of the progeny of these progenitor cells are going to be expressing the transgene that you put in the retrovirus. If we put GFP, we can just visualize the cells. So this is a single injection of GFP in an adult animal that received this injection. We sacrifice the animal several weeks later, and then we can have these green cells, which are adult-born neurons, extending dendrites towards the molecular layer and axons toward the hilus towards CA3. So here you can see that these cells are basically covering, and these are only a few cells that are being labeled, covering the molecular layer with dendrites of adult-born neurons, and then coating the hilus and CA3 with axons of adult-born neurons. Yes? This is the dorsal hippocampus. The ventral hippocampus is the same. So the septotemporal axis is very, very similar. The developmental process is a little bit different. It's much slower in the temporal hippocampus, but the whole phenomenon is the same. So the point that I was mentioning is that these cells are receiving GABAergic connections in the dendrites and perisomatic connections in the soma, but then the axons are contacting cells on the way. And one way to ask the question is, of course, who is connected to these adult-born neurons? One way is to do physiology by doing whole cell recordings, for example, in a green cell, and then going blind to look for interneurons in the postsynaptic area and see if you can find synaptic pairs, or actually use a retrovirus to express channel rhodopsin, and then having channel rhodopsin in many cells at a time in the dentate gyrus, you can basically do slices, go to record postsynaptic cells blind, but then when you flash a blue light, you can activate all of the neurons in the presynaptic area and then look for the target, who are connected, who are the target cells of these neurons. Question that we try to address in these experiments, and the experiments were done by Silvio Temprana, Lucas Mongiat, and Sanming Yang, sitting here in the audience, is, so what are the different targets to these cells as they develop in the network? And we are going to call young cells neurons that are being born in the adult and are 4 weeks old, 4 WPI. And mature neurons we're going to call cells which are 7 or 8 weeks old. And we are going to ask who are the targets to these cells, okay? So these cells are now channel rhodopsin expressing retrovirus is injected in the dentate gyrus. Animals are sacrificed at 4 weeks or 7 weeks. We make slices, we put them in the microscope, and then we record postsynaptic currents activated by a flash of blue light. First of all, we have to see that when we flash a blue light, then you can activate the green cells. Because one point that has to be taken really carefully with optogenetics is that it's not the same as stimulating a neuron with an electrode in which we know, or you know, that when you give an electrical stimulus, the cell is going to fire 1 or 2 or 3 action potentials depending on how many pulses you are giving. But in terms of channel rhodopsin stimulation, you are activating a channel that has a particular kinetics for activation and inactivation, and you can get conditions in which one flash of light is going to give you 3 or 4 spikes, and you need to make sure that when you give a flash of blue light, you can assume that the cells are firing one single spike, and then you're going to address what's happening after cells are firing one spike. So in this case, first we are checking with cell-attached recordings here by shining a blue light in the slides and putting a loose patch pipette in the cells that each time we flash a blue light for 1 millisecond, then the cells are making one spike. And this is the same case for young cells and for mature cells. And now that we know that when we flash a blue light, there is one spike, we are going to ask, what are the rest of the network seeing? First, we're going to ask a question of what happens when we stimulate young or mature neurons when we patch pyramidal cells in CA3. And the circuit we are looking at here is a direct connection between the granule cell and the CA3 pyramidal cell. And also, we may get feed-forward inhibition by GABAergic interneurons in the hilus or in CA3. So now we flash a blue light, channel rhodopsin is in granule cells, we are recording from pyramidal cells. And by doing an electrophysiological trick, if we do recordings at minus 70 millivolt, we are going to record excitation. This is an inward current, and this inward current is unaffected by pyrotoxin, which is a blocker of GABA receptors, and is blocked by kynurenic acid. So this is telling us this is a monosynaptic excitation. And if we record at the reversal potential of the excitation at 0 millivolts, so there's no glutamatergic currents, and we have only GABAergic currents, here we can find feed-forward inhibition, and feed-forward currents are blocked by pyrotoxin, but they're also blocked by kynurenic acid, and this is showing that we are blocking this excitatory connection and this inhibitory connection. So this is a desynaptic feed-forward inhibition, and this is a monosynaptic excitation. Now the question we want to ask is, what happens when this presynaptic cell is a young 4-week-old neuron, or when this presynaptic cell is a 8 or 7-week-old neuron, which is a fully mature cell? So when we stimulate young neurons, we see that there is inhibition, feed-forward inhibition, and there is excitation. If we stimulate presynaptic mature neurons, we find also inhibition and excitation. And if we quantify the amount of inhibition and the amount of excitation, and we compare, here we are excitation in CA3 pyramidal cells, when the presynaptic cell is young or mature, it's pretty similar. So this is telling us that 4-week-old neurons are already reaching target cells in CA3. In terms of feed-forward inhibition, there is a little bit stronger inhibition in mature neurons, but basically there is no significant difference. So 4-week-old neurons are already making mature contacts in CA3, both on GABA interneurons and on pyramidal cells. So what happens with the feedback inhibition? So now what we are doing is a similar experiment. We are stimulating granule cells, and we are recording from neighbor granule cells. So what we are going to be looking at is the inhibition that is activating by young or mature granule cells onto neighboring granule cells. And here you can see that when the presynaptic cell is young, inhibition is very weak, and when the presynaptic cell is mature, inhibition is very strong. So this is actually an experiment that was a bit surprising for us, because what this is telling us is that even if axons initially, of course, grow through the hilus and then reach CA3, the CA3 layer, so initially our hypothesis was, okay, if there is any difference, they are first initially going to contact hilar interneurons, and then later they are going to be contacting CA3 targets. But what we find is that they initially contact CA3 targets, and there is a delay in the GABAergic, in the output synaptogenesis towards GABAergic interneurons in the proximal area. So basically, these cells are activating feed-forward inhibition and CA3 pyramidal cells, and they fail to activate feedback inhibition that is going to control granule cells in the granule cell layer. So far, yes?
Did you try to record newborn cell as postsynaptic?
Yeah, we did all the conditions. So the question was if we recorded newborn cells as a postsynaptic cell, and the answer is yes. And I didn't put the results here, but they also receive very weak inhibition when they are 4 weeks old. So there is both. They recruit weak inhibition and they receive weak inhibition. So the full coupling to inhibition is delayed. Okay, so we wanted to check the importance of this feedback inhibition to really control the spiking of the granule cell layer. And this is an experiment that Yang did here. And the idea here is that we have the network in which cells are receiving excitation from the perforant path from the entorhinal cortex. We stimulate perforant path in the slice. And then we put an electrode to do field recordings. And with the field recordings, we are going to be listening to the activity of many dentate granule cells in the same layer. Okay? And the type of recording that you get here is a very strong depolarizing signal, and then a peak. And this peak is going to present the population spike. And this population spike is the spiking of many cells in the granule cell layer at the same time. So if you stimulate strong enough these axons, many of these granule cells are going to spike, and then you're going to get the population spike in the signal. And now the question is, if we pair this stimulus with a flash of blue light to activate feedback inhibition, are these cells able to control the firing of the granule cell layer? This is the question of this experiment. So this is an example of experiment in which in one we are flashing the blue light, in two we are going to stimulate the perforant path, and then we get the population spike. And when the flash of blue light is getting closer to the stimulation of the perforant path, at some point the population spike is getting smaller, and then when the blue light goes closer and closer and then it's too late, then the population spike is recovered. So this is showing us that by flashing a blue light and activating a very small population of granule cells that are labeled here expressing channel rhodopsin, this is enough signal to give feedback inhibition to the granule cell layer to silence the rest of the granule cells. So now the question is, okay, this is the case for mature neurons. What happens when the presynaptic granule cells are immature? And basically the answer is there is much weaker inhibition when presynaptic cells are young than when they are mature. And this is consistent with the fact that the young neurons are recruiting poor feedback inhibition. And this is an experiment basically showing that this effect depends on picrotoxin. If you block GABAergic connections with picrotoxin, then there is no reduction of the population spike induced by light. So these experiments are showing us that young granule cells born in the adult hippocampus are exerting poor control of the granule cell layer, and the population spike reductions that we can see induced by mature cells really involves feedback inhibition. And there is an experiment that Yang has done a few days ago, and I think it's... What Yang did in this experiment is, instead of expressing channel rhodopsin in the granule cells now, he expressed channel rhodopsin in the parvalbumin expressing interneurons, in the GABAergic interneurons. And this is done by using a transgenic mouse that has the Cre driver, the Cre recombinase under the parvalbumin promoter, crossed with a mouse that is expressing channel rhodopsin with a flock stop cassette. So then by crossing the two mice, you have all parvalbumin cells expressing channel rhodopsin now. And now by flashing a blue light, now you are going to activate all of the interneurons that are present in that slice. Okay? So this is activation of parvalbumin interneurons by channel rhodopsin. And now Yang did the same experiment. He stimulated the perforant path and recorded the population spike. And this is the type of signal that you see. And this is the type of signal that Yang was finding. And then by increasing the laser power from 15%, 18%, 22, and higher, here you can see that the population spike is finally completely abolished. So this is showing us that parvalbumin interneurons can be mediating the effect of feedback inhibition from granule cells onto the granule cell layer. So one of the strengths of this technique of optogenetics is that you can target your tools, the channel rhodopsins or allorhodopsins or whatever you want to do to the cells, to specific populations of neurons, which in some cases you might be able to stimulate them with electrodes, but in this case it's impossible really to stimulate all GABAergic interneurons in a tissue by any other technique which is not this genetically directed expression of these controller proteins. Really briefly, we also use another technique which is called now chemogenetics, but what it basically is the use of synthetic receptors that have been modified to, these are ligand-gated receptors. In this case, they are G-coupled proteins, and these G-coupled proteins are modified so they don't bind their typical ligand, but they're going to bind a synthetic ligand. In this case, we are going to work with the HM3, which is a human muscarinic receptor that instead of binding acetylcholine is going to bind clozapine N-oxide. These are receptors that have been developed by Brian Roth in North Carolina. And these are really cool tools because they allow to control neuronal activity now, not by flashing a blue light, which in a way is also the disadvantage of that method is that it's invasive. You need to put an optic fiber and you need to be able to provide the blue light inside the brain. But in this case, you can basically use a ligand that you can inject in the animal, not in the brain, but systemically now, and activate the receptors that might activate neurons in case of the human muscarinic HM3 receptor, or inhibit neurons in case of the HM4-DI receptor. So here we have two tools that we can also express in different populations of cells and control their activity by injecting the ligand into the animal. Of course, the time course of experiment is totally different because here you need to have a ligand that is going to reach the brain and activate the receptor, so you cannot really control spike by spike the neuron, which is the case for channel rhodopsin, but you can activate the neuron for a while during a given time frame. And the experiment we try to do now is, okay, I showed you all the connectivity of these dentate granule cells born in adult hippocampus in slices, okay? Can we prove that something similar is occurring in the intact brain in the live animal? So for that experiment, what we did is we used the HM3DQ, which is going to inhibit GIRK channels, so it's going to depolarize neurons, so it's an activator of neurons. We put this receptor in a retrovirus, we injected in the dentate gyrus, and now we are going to have adult-born neurons that are going to express the HM3DQ receptor, okay? So adult-born neurons now can be activated by CNO, and these are experiments that Mariela did, who is sitting here in the audience. So the question is, we now activate neurons at different ages, 4 versus 8-week-old, and we want to know what target cells are being activated downstream. And we are going to direct the question directly to parvalbumin interneurons in this case, because are the main suspects of the neurons that are responsible for the feedback inhibition. So with this expression and the experiment we ask is, if we activate 4-week-old neurons or 8-week-old neurons, can we see parvalbumin interneurons that express C4s, which is a marker of activity there? The summary of the experiment is here, but I'm going to tell you how we did it. So we are going to compare parvalbumin expressing C4s interneurons in the ipsilateral hemisphere to the contralateral. So this is the same brain of the same animal. And here we can find that when the presynaptic cells are mature neurons, there is a much higher expression of C4s in PV-positive interneurons compared to the contralateral brain, which is not the case for young neurons. So this is showing us that mature but not immature cells in vivo are activating parvalbumin interneurons in the hilus. So this is a way to show that this technique that a priori feels that it's not as powerful as optogenetics can allow you to address a completely different question, because in this case we are not doing any invasive treatment. We just inject the drug and we wait for 3 hours and then the PV cells are on, okay? This is the people that did experiments. I'm sure you made many data by Yang and also by Mariela here and also by Silvio, Lucas Mongiat, Antonia Marin-Burgin. And thank you very much for being here.
Is there anyhow to control the amount of RNA that the virus will be producing in the cells? Because this will change the response to the light.
Well, the virus is not producing RNA. Actually, the virus is being integrated into the genome of the cell. And of course, with the retrovirus, you're going to have insertions in different places and you're going to have variability in the population. And this is something that you can't really control, so you have different cells with different expression levels. But if you sample enough cells and you make sure that your stimulus is the same for the different conditions that you are comparing, let's say 4 versus 8 weeks old, then you are going to have a heterogeneity in the population, but as long as you control it, it's fine. And if you use the genetic approach, it's even better because then the expression sites for the transgenes are always the same, right? When you use the Cree recombinase to remove a flock stop cassette. Yes. I think that's even a better technique. We are combining both things and even labeling adult neurogenesis using the genetic approach.
Okay, thank you.
I think I remember your talk in the last Ibro in Florencia,
wow.
More or less, but I think you did the pair recording instead of optogenetics.
No. We never did pair recordings. I mean, there's no graduate student that would ever do this experiment for me.
Okay, okay. But my question is, if you have some comparison between the, I don't know if not a pair recording, but my question is, when you insert this optogenetic tools, is there any phenotype change of this
we
granule
checked a long
cells?
time ago. We checked at the basic intrinsic properties and synaptic connections and excitation, and it's the same. It doesn't seem to be changing.
Yeah, and another question is, I know these cells has this transmitter switching system. So could we interpret your data also if you increase the blue light power, a lot of excitation of these granule cells, is it possible that these cells switch transmitter from glutamate to GABA? Then you have the inhibition.
I mean, well, first of all, we are doing acute experiments, so we are just doing this within tens of minutes. But once they are glutamatergic, I don't think you are going to switch them to GABA. I mean, there is some evidence that they may co-release in some places GABA. But with this brief stimuli, you're not going to change the phenotype of the cells.
I was wondering whether you've done any electrophysiology in slices to look at the effect of CNO and HM3 activation. Because one possibility might be that maybe...
We did that, and we got some... It's not great. It's not something that you can just puff it in the bath and you can see spikes. What we did in vivo is to inject the CNO and look that those cells are lighting arc like crazy. But to look at the activity is not so easy. So we are actually switching from these tools now to Scott-Sternsson's activator and inhibitor, which are ion channels, which are activated by synthetic ligands, and then you can control them much better. So we're actually moving away from the G-coupled receptors towards ion channels. But I mean, that's a good point. You don't know exactly what's happening in the cell. Well, thank you very much. Our next